Hi, everyone, and welcome to my session today on the limb and vertebrate development. My name is Harry Miles, and I'm a lecturer in anatomy. So, the intended learning outcomes of today, we're going to look at understanding the development of the limbs and spine, and then after that, we're going to understand how congenital anomalies can arise in the limbs and vertebra. Now, as a little rough session plan, we're going to have a look at development of the limbs first, specifically looking at how the limb actually elongates, how a ulnar and radial axis is formed, then how the digits are formed, and then a bit of pathology. After that, we're going to look at the development of the spine, specifically the neural tube, vertebra, and pathologies associated with it. So, in terms of development of the limb, the general idea is that the embryo induces something called the limb bud, which we can see here. I'll circle it just there. And then this will more or less just go on to become the limb. Now, when we talk about the limbs in the embryo, we don't tend to use the words upper limb or lower limb. We tend to use the words forelimb and hind limb. And this is because if you think about how us as humans, since we walk on two legs, it kind of makes sense that we say, well, we've got two upper limbs and two lower limbs. But once upon a time, we were on all four legs. And there's a lot of animals that still are on all four legs. And so it makes more sense today to say that they've got two front limbs and two back limbs. And this is why we have the forelimbs and the hind limbs. So in today's session, I'm going to focus a little bit more in terms of the upper limb. So I'm going to refer to the forelimb a bit more, but the same sort of principles will apply to the hind limb. So this is just a nice sort of brief overview of how the human embryo, so from about sort of 5 weeks or so, the limb bud forms. You have the outgrowth and the patterning. The patterning sort of tells you here what these are going to go on to become. Then we have the growth and the differentiation, and this is where we get this sort of typical osteology that we tend to see. Now, we actually refer to the limb in 3 different regions, essentially. So we have the stylopod, which is in reference to the humerus or the femur. We then have the zygopod, which is in reference to the ulna and the radius, or the tibia and the fibula. And then we have the orthopod. Now, the orthopod is talking about the wrist, fingers, the ankles, and the toes. And these are all very well conserved. Just some examples I've got up on the screen here. Just here, we've got a chick forelimb. So we can see here, we've got the humerus, ulnar, and radius with our stylopod and the zygopod. We then have the metacarpals and the digits in the orthopod. We then look down here at the mouse forelimb, which looks a little bit more like a human in that sense. You know, we've got that stylopod here, we've got the zygopod bones here, and then we've got the orthopod here with sort of the carpals and the metacarpals. Now, one of the quite interesting ones that I find quite fascinating is things like the bat forelimb. So when you look at a bat, you just sort of see these wings, but if you were to look at the osteology, you can see how it's got, well, that's the equivalent of the humerus. So this is our stylopod. We've then got a radius and an ulna. So that's going to be our zygopod. But then the actual part that forms the wing, you can see there's sort of 5 distinct digits. However, they're a lot longer than our fingers, shall we say. And so we also have the orthopod here. Another quite interesting comparison is that of the horse. So the horse, if we sort of just take it from this point here, but over here we've got the radius and ulna, which is, as you can see, it's just more or less one bone. It's sort of fused together. But we then have the carpal bones, followed by metacarpal 3, and then it finishes off with just a distal phalanx. And so what we can sort of see going on here is that digits 1, 2, 4, and 5 have sort of gone away. They've sort of been lost over time. And so the horse more or less just walks on its finger, which you can sort of see as it's compared to a human's finger here. So horses literally run on their middle digit, which is kind of weird, but actually kind of fascinating too. Now, just in terms of some areas of importance. So here we have that limb bud, which we can see coming out here. Now, just like in when we talk about our limbs, we have a proximal and a distal, and it's done in the exact same way. Proximal right next to the trunk, distal moving away. So that would make this part here our proximal and this part here our distal. And so at the sort of distalmost part of the limb bud, we have a region of cells called the AER, or the apical ectodermal ridge. Now, the AER is actually responsible for progressing the growth of the limb itself. It produces something called FGF, which is used in quite a few different signaling pathways, which we'll get on to a little bit later. We've then got the progress zone. And now the progress zone, or PZ, this is a sort of rapidly dividing mesodermal cells that essentially cause elongation of the limb. So it's these cells here that are more or less actually causing the elongation or the movement more distally of the limb bud. Now, the final part that I wanted to talk about is the ZPA, or the zone of polarizing activity. Now, the zone of polarizing activity, or ZPA, is responsible for determining the ulnar radial axis in the forelimb in this particular case. And it produces something called SHH, or sonic hedgehog, which again we'll sort of get into a little bit later. But first of all, let's go back to AER, so that apical ectodermal ridge. It produces FGF. And so if the AER is intact throughout development, what we tend to find is a very sort of normal, a wild type limb being produced. However, if you were to remove the AER early on in development, then we will only be left with the humerus in this case. And then if we were to remove the AER a bit later on, we have the humerus, but then we also have the ulna and the radius. Now, this doesn't just necessarily refer to the fact that the AER is being removed. There could be something just disrupting the FGF signaling. An example of this is something like thalidomide, which again I'll touch on in a second. But here's an example of where either the AER could be removed, or if FGF signaling is impacted quite early on in development, what will happen is the limb just will not form. And so the way that you can think about it is like the AER being removed a lot earlier than this point here. Or the FGF signaling being impacted a lot earlier than this part here, leading to basically a malformation of the limb, or no limb formation, should I say. And this is called amelia. Okay. Now, we've got the opposite side of that. And if the AER, or the damage, or if there is an impact on the FGF signaling a bit later on in development, so later on than at this stage here, what can happen is we just have maybe one digit go missing. And so what we've got here is a very sort of Simpsons looking hand type thing, or very cartoonish sort of hand that's missing a digit. And this is called adacyly. Now, moving on to ZPA, or the zone of polarizing activity. So the red sort of circles here are representing sonic hedgehog, or that SHH, which is a mutagen. The way that I always like to think about it is, if you've got the ZPA producing lots and lots of sonic hedgehog, in the cells surrounding the ZPA, there's going to be a much higher concentration of sonic hedgehog. And then as you move further and further away from the ZPA, the cells are going to have a much lower concentration of SHH, or sonic hedgehog. And in this particular case of this mutagen, the cell's fate is determined by the concentration of sonic hedgehog it's exposed to. So just as an example, this bunch of cells here that are right next to the ZPA, they're going to have a different cell fate to the ones that have got a low concentration of sonic hedgehog much further away. Now, I appreciate that this is kind of a hard concept to get your head around, and so I've sort of tried to put it into another couple of different examples, but just first, the high concentration of sonic hedgehog, in this case here, leads to sort of ulnar structures being developed, whereas up here, where there's a lower concentration, it leads to radial structures being developed. And I like to sort of compare this to as if we've got here a ZPA person who's currently holding up a rope. So the rope in this case is actually representing the concentration of sonic hedgehog. Okay, so here we have the ZPA person who right next to it has got the highest concentration because it's producing it straight away. And then what we have here is we have got 1, 2, and 3 people at different distances from the ZPA person. So we've got person number 1 that's a lot closer to ZPA, and we've got person number 3 who's much further away from ZPA person. And if we were to follow this rope, so we're now looking at the concentration gradient, in line with number 1, we've got the head. The rope crosses over in front of number 1's head. The rope then crosses over at number 2's sort of belly, and for number 3, it crosses over with their leg. And so what we could say is, at this concentration here, we would make the head. At this concentration here with number 2, we would make belly. And all the way down here, where there's a much lower concentration, we would make a leg. And so this is pretty much what happens here. What we have is, where there's a very low concentration, we have the formation of digit number 1 all the way through to digit number 5. And this is all to do with the concentration of sonic hedgehog that it's exposed to. Now, technically speaking, the cells exactly here and these cells exactly here may not actually go on to become the digits. It's more just a general way to help you identify what's likely to be occurring and where. However, when we do start talking about digit formation, it's kind of important to think about how, looking at just sort of the limb bud, there's no distinct sort of digits here. It's quite literally just a bud in that sense. And so in order for us to get to this stage here, where we have the sort of distinct fingers, we need to have apoptosis, or a programmed cell death, take place, sort of going between the phalanges. And hopefully this sort of, in your head, gets you thinking that, well, if this is the normal digit formation, then you can kind of see how digit malformation can also happen. So if the apoptosis doesn't actually occur, what we have is a fusion taking place of the digits. And this is something called syndactyly. Now, this is obviously quite a severe form, as two fingers are quite literally just attached to each other. But if we look over here, this is just an example where we have someone with webbed fingers. And again, you may have seen people who have webbed toes as well. And this is just quite literally because the apoptosis hasn't gone as far down as maybe it should have. Now, moving on to development of sort of the neurotube in this case. So here we've got the neural plate. Now, the neural plate pretty much just folds up on itself like this. It forms this nice little neural groove here. But then what we have is, at this sort of convergence point, is a nice sort of sealing up here, forming a nice tube. Okay. And that's why this is the neural tube. Okay. So it's a tube just full of nervous tissues, essentially. And over the top of this, we have the epidermis. Now, there was a few structures missing from that picture before, which I've added in myself here. So just about here, we've got the notochord. And then more or less sort of either side of the neurotube, sort of going all the way up and down it, we have the somites. Now, the somites are very important for many different reasons, but in terms of the development of the sclerotome, they are very, very much quite vital. Now, the somite will more or less split into these two parts. You have the dermomyotome, and then this will further split later down the line into the dermatome and the myotome, and then also into the sclerotome. Now, the sclerotome, these will actually go on to form bone, essentially. And it does this through sort of forming chondroblasts first, and then forming the hard bone that we associate with the spine now. What we see happens is the sclerotome, at a certain stage in development, will sort of split away from the dermomyotome, and it will surround itself around the notochord and the neural tube. And apologies, I'm not exactly a great animator, but it'll look a little bit like that, essentially. So we have a sclerotome more or less surrounding the neural tube and the notochord around here. Now, this is another schematic, but it's a little bit more accurate than this one. But here we've got a really nice picture that sort of shows how we go from the sort of the somite over here, and then it sort of slowly progresses. You see the sclerotome sort of surrounding the neural tube and the notochord until we get to this sort of point like this. And now, in terms of development of the vertebra, we're now looking at the neurotube on a slightly different axis. So before we were sort of looking at it from a sort of cranial view almost, or it could be a caudal view as well. And we're now sort of looking at it from maybe an anterior point of view or a posterior point of view. And what we have is we have the sclerotome shown here, sort of sitting either side of the neurotube like this. However, we've got some myotome sort of sitting just next to the sclerotome, which again we see sort of around here as well. And what happens is you have part of the sclerotome actually move cranially, and then you have another part of it move caudally. And what this means is that you have one part like this moving down, this part here moving up, and it essentially allows for these nice little branches of spinal nerves to come out and move between these little fissures. Now, these fissures are called von Ebner's fissures. And these essentially allow for the channel for the spinal nerves or the cranial nerves to actually get to the myotome, which will go on to become muscle. And so when we start looking a bit later on, you can see how you have the nerves now actually already at the muscles. And then what we can have from here is wherever these muscles end up going, whether it's right into the hand or right into the feet, these nerves can actually follow with it. And what you don't have is sort of nerves getting lost and trying to find the muscle. Instead, just we have the muscle and the nerves develop quite close to each other, and then they move off with each other. Now, referring back to the sclerotome here, we had that part that was moving caudally. We had that another part moving cranially. These two parts will actually fuse together, and they will form vertebra like this. And so this is another picture that sort of just shows how that works. However, now we need to start thinking about how this relates also to the notochord. So obviously this bit here was just talking about the sclerotome surrounding the neural tube here. We're now going to start talking about the sclerotome surrounding the notochord down here. And so here we have that sclerotome surrounding that notochord. And so the same thing's happening. We have one part of the sclerotome moving caudally. We have another part of it moving cranially. This part of the sclerotome is fused, and this will actually go on to become the vertebral bodies. And then what we also have is we have another part of sclerotome that is sort of between these vertebral bodies, which ends up becoming the intervertebral disc, which we can sort of see just sort of gets passed forward like this. And so this intervertebral disc does not end up becoming bone like how these parts here do. This becomes this sort of fibrous, almost gelatinous structure called the annulus fibrosus. And this is sort of just acting as the shock absorber for the body. However, when we then start thinking about the notochord, which is still traveling down the center here, the notochord now is going to be sort of seen in the center of these intervertebral discs. And so what these end up becoming is something called the nucleus pulposus. Now, just checking in with a few of the anomalies that can happen. So here we've got an example of something called hemivertebra. And so this is a vertebra that has quite clearly sort of formed on one side, but it hasn't formed on the other. And just one of the possible explanations for this is that the somite here has been absolutely fine on one side, but on the other side, maybe it hasn't. And this could be for any number of reasons. And to be fair, if there's no somite on the other side, there's probably a few more problems other than just this hemivertebra that's going to be seen. But another thing that might happen is that maybe there's a lack of blood supply to this part here, and so it dies. And again, this is all just sort of things that can happen by chance or be a congenital anomaly. Another one we have is fused vertebra. So hopefully you'll remember from earlier on, we had some parts of the vertebral bodies wanted to become bone. So again, these become chondroblasts, and then this goes on to become bone. However, the intervertebral discs don't do that. However, as you can see, on these three here, for some reason, the intervertebral discs on this half have actually ossified. They've become bony. And so what this has done is it's essentially just fused these vertebrae together, which can be quite painful. And there's going to be a whole other load of reasons as to why this can actually happen. One of the ideas that I always like to sort of think about is it gets too excited and it starts thinking, oh, yes, turn everything to bone. Instead of for the intervertebral discs, they're sort of, saying, you know what, chill out, it's fine. Another one we have is something called butterfly vertebra. So here we have a normal vertebral column, and here we've got something called some butterfly vertebra. And there's quite a few of them that you can see here. And essentially, it's to do with the failure of the fusion of the vertebra in this sort of sagittal plane. So you can almost call it a sagittal cleft in that sense. And for some reason, they haven't fused together in the sagittal plane. And so that's all from me. Thank you so much for listening. In terms of further reading, I'd recommend looking on nervous system and the developing human. Anyway, my name's Harry Miles, and that's all I've got for you today. See ya.